Thank you for the invitation. Yesterday at the networking dinner, several people asked me, what does obsidian mean? So obsidian is formed from volcanic eruptions. It's a volcanic glass, and it's used to make really sharp blades. So the reason we chose that name is that we're at the bleeding edge, way ahead of the cutting edge of technology. More specifically, every single volcanic eruption has a unique fingerprint to the obsidian that is made. Hence, we are personalized and at the cutting edge of technology. I'm Madan Jagasia, CEO of Obsidian Therapeutics. Our mission statement is we are a clinical stage biotechnology company developing a next generation, regulatable, armored, tumor-infiltrating lymphocyte cell therapy for solid tumors. Most of my presentation today is going to be about a lead asset, OBX-115, and this is a first-in-class genetically engineered TIL cell therapy where we are pharmacologically regulating membrane-bound IL-15. Our executive team has significant experience, not just in oncology or cell therapy, but more specifically in TIL cell therapy. This expertise goes all the way to the board chair, Dr. Maria Fardas, former CEO of Iovance, and as you probably heard yesterday in the panel discussion, Iovance recently had their product approved in the United States for post-checkpoint metastatic melanoma. We have two ongoing research collaborations with strategic partners, one with BMS, the other with Vertex, where we use our core platform, CytoDrive. I'll talk about that in the next few slides. BMS is leveraging that platform to regulate CD40 ligand in an oncology indication, while Vertex is using that to regulate a gene cassette in a non-oncology indication. We are well capitalized. In fact, just last week, we announced that we completed our Series C financing. We were oversubscribed and raised $160.5 million, backed by premier investors. We are active in the clinic. We have 3 clinical trials ongoing. I'm going to be sharing some data from the first study. The first chevron that you see is OBX-115 in metastatic melanoma. This is a single center study ongoing at MD Anderson. And the drug product OBX-115 is manufactured at Cell Therapy Manufacturing Center, or CTMC, which is a joint venture between MD Anderson and National Resilience. We have a multi-center study ongoing. This is under the Obsidian IND. One cohort is in melanoma, and the other cohort is in non-small cell lung cancer. So before we go into the clinical data, let's zoom out and ask ourselves, why TILs? The challenge with solid tumors is that these tumors have a diverse array of neoantigens. Many of these neoantigens are not shared across patients. Yesterday we heard that some antigens are shared across patients, but then these are HLA restricted as you try to convert these to TCR-T therapies. If you take the tumor, you can coax the TILs out during manufacturing. And the TILs have already achieved 2 fundamental things. They recognize the neoantigen, they're polyclonal, and they've already trafficked to the tumor microenvironment. So you can convert this to a cell therapy. This is the foundation of non-engineered TIL cell therapy. Unfortunately, for non-engineered TIL cell therapy, there is suboptimal persistence and expansion of the cells, and there's always going to be a limitation in the efficacy. At Obsidian, we are leveraging the CytoDrive technology to regulate membrane-bound IL-15 because it can fundamentally solve both expansion and persistence and should translate into improved efficacy. The other challenge with non-engineered TIL cell therapy is that in order to grow them, you have to expose them to high doses of IL-2. IL-2 does a great job at expanding T cells, but it comes at a price. These T cells are now terminally differentiated and they're exhausted. Once the T cells have seen IL-2 in culture conditions, they're addicted to IL-2, and now you have to give high-dose IL-2 to the patient, which leads to a domino sequence of events, including the side effects of IL-2, and you restrict your market accessibility, and the safety profile is not the greatest. So at Obsidian, our thought process is, if we can regulate membrane-bound IL-15, we get away from IL-2 during the TIL expansion and manufacturing. We don't require IL-2 in the patient, so we should have a differentiated product and a safer product. We try to quantify this in this slide. So let's imagine you have 100 patients who medically need TIL cell therapy, depicted by the 100 humanoids on the left. In order to get unengineered TIL cell therapy with high-dose IL-2, the organ functions need to be pristine. The patient needs to have a normal heart function, lung function, liver function. When we talk to KOLs in a market research, they feel that only 20 to 40% of the patients who need TIL cell therapy can actually get the unengineered TIL cell therapy. What's the impact on these patients? We know that the current approved product has a black box warning for treatment-related mortality. This product has to be given in the inpatient setting along with intensive care unit support. And there's a huge health economic impact because taking care of patients in the ICU is not cheap. Now let's think about OBX115. Our product does not require IL-2. We can manufacture the product with a core needle biopsy. We believe that our product is compatible with lower doses of lymphodepletion. If you line up all of these differentiators, the key opinion leaders believe that OBX115 should be able to cater to 80% of the patients who medically need TIL cell therapy. A few words on our platform, CytoDrive. So in the case of OBX115, our gene of interest is membrane-bound IL-15. This is brought under the control of a drug response domain. For OBX115, the DRD encodes for a carbonic anhydrase sequence. This is transfected into the TIL cells. The ligand for the DRD is an FDA-approved small molecule called acetazolamide. If you've climbed mountains, gone to high altitudes, you've probably popped this drug called Diamox or acetazolamide. It's innocuous, it's used as a mild diuretic as well, and for open-angle glaucoma. In the absence of the ligand, the fusion protein of the DRD and membrane-bound IL-15 is degraded by the proteasome, and that's what we call the drug-off state. When you expose the cells to the ligand, either in manufacturing or give acetazolamide to the patient, you get stabilization of the DRD and IL-15 construct, and now you get cell surface expression. So rather than thinking about this as a binary off state and an on state, think about this as tunable, because the expression of IL-15 can depend on the dose, duration, and frequency of acetazolamide. Now let's look at the mechanism of action, walking from left to right. So the patient gets lymphodepletion, gets the drug product OBX115, gets the ligand ACZ, the DRD is engaged, IL-15 is on the cell surface. The way the construct has been engineered, based on the linker length, the membrane-bound IL-15 can loop over and signal through its own IL-2 gamma receptor, or it can present that signal to a neighboring immune cell. On the right-hand side, what you see is the downstream effects of normal IL-15 signaling, namely antigen-independent expansion and persistence of the TILs. The TCRs now recognize the neoantigen. You get direct killing of tumor cells. And the other important aspect is, during the IL-15 signaling, you get memory TIL formation, which drives the persistence of the product, which is really important in the world of T-cell therapy. In the transactivation, the IL-15 signal can be presented to a neighboring natural killer cell or NK cell that is recovering from the effects of lymphodepletion. These NK cells, now we have translational data, can actually expand in the patient, traffic back to the tumor. And this may become really important in a disease like non-small cell lung cancer, where in 20% of non-small cell lung cancer, post-checkpoint, the mechanism of resistance is downregulation of class 1. If you're downregulation of class 1, your TCRs cannot see the cell, but the NK cells is a natural target for those neoplastic cells. A few words on manufacturing. The first phase of manufacturing is called pre-rep or pre-rapid expansion protocol. We have an ancillary drug substance in this step that is not present in the non-engineered platform. And this drug substance allows us to selectively capture the T cells in the tumor microenvironment that have recently been activated. This is the reason we are able to make the drug product with core needle biopsies and do not need a surgical excision. The activation and transduction is standard. This is with the retrovirus. In the rep phase, we have a lot of differentiators compared to the non-engineered process. Because our transgene is already in, we add the ligand acetazolamide, which drives the IL-15 and leverages the IL-15 to drive the T cell expansion. In this process, the T cells need a co-stimulatory signal. This is provided by an engineered feeder cell, which we call I-feeder cell, and these feeder cells now express IL-21 and 4-1BB. So the entire process is tailored to the end product, which is CD8 enriched, minimally exhausted, having stem-like characteristics to drive further proliferation and persistence. So as we think about OBX115, what I'll be showing you in the next few slides is that we have the ability to make the drug product by minimizing surgical excision. OBX115 is potentially compatible with lower doses of lymphodepletion. We are IL-2 sparing, and if you line up these three variables, can we take TIL cell therapy completely to an outpatient platform? You'll be seeing clinical data where we can show that we have rapid onset of tumor control. We have the biologic rationale to redose acetazolamide several weeks after the infusion to deepen the response and further extend the persistence of the cells. We already have data in our early data set that we can see complete remissions. So when you combine the transformational outpatient play and the efficacy, you can now speculate that this has a blockbuster potential and can cater to the unmet need in a comprehensive way. This is the schematic of the first-in-human study. The tumor is collected, comes to us for manufacturing. There's a baseline assessment, lymphodepletion, product, ligand, and efficacy and safety. Just a reminder, there is no IL-2 in the regimen. We presented data at AACR yesterday. The first 6 patients, now updated to 25 weeks, so 6-month follow-up. All patients are anti-PD-1 primary resistant. That means their best response was progressive disease. All patients had received anti-CTLA-4 and were resistant to that. Out of the 6 patients, 3 have responded for an ORR of 50%, including 2 CRs, no primary progressors, giving us a DCR of 100%. The safety profile is differentiated. There were no DLTs, no grade 4 non-hematologic events, no patients to the intensive care unit. The translational data that we have has confirmed the platform validation. Here is the swim lane plot, which was shown at AACR yesterday. Let me talk about patient 1. This is a young patient, primary resistant to anti-PD-1 and anti-CTLA-4, multiple lines of therapy, no response, multiple surgery, multiple radiation, brain lesion that was stabilized with radiation and then comes to OBX115. Week 6, PR, week 12, PR, week 12 CR, which has now been sustained for more than a year. What we learned from that patient is that we have the opportunity to redose acetazolamide. We had that amendment for patient 2, who decided not to get the redose, stayed with stable disease through week 24, and then had a progressive disease. This patient started standard of care, has again gone into a near CR. Patient 3, 4, 5, and 6 have all seen a redose of acetazolamide, and as you can see in patient 3, they had stable disease till week 6, got the redose of acetazolamide, and then moved into a CR at week 18, while patient 5 got a redose of acetazolamide and achieved a PR at week 12. So what's our vision? Currently, we've delivered on our first-in-human data. We have a multi-center study ongoing both for melanoma and non-small cell lung cancer. We've heard loud and clear from the FDA, engage early, engage often, right? We know that the potency assay for unengineered TIL cell therapy has been a challenge, but we believe that potency assay for OBX115, where the mechanism of action is anchored around IL-15, is easier to prove to the FDA versus an unengineered TIL cell therapy. We see a direct path to registration for melanoma. We are now in non-small cell lung cancer and working on our next generation assets. How are we thinking about our next generation asset? OBX115 with IL-15 engineering has optimized the cell therapy characteristics. We have the ability using cytodrive to control pleiotropic and potent cytokines. So we could technically add IL-15 plus cytokine X, where cytokine X's role is to modulate the tumor microenvironment so we get the synergy between IL-15 physiology and the new cytokine. We also believe that the membrane-bound IL-15 and DRD, if we put it into a CAR-T construct or an NK CAR construct, could that potentially get rid of lymphodepletion? Because the focus of lymphodepletion is to drive the ALC down so that you get an increase in the endogenous IL-15. Our product makes its own IL-15 on demand. So we are asking these theoretical questions currently and gradually moving ahead to develop these platform collaborations. Thank you for your attention.